videos, we've talked about the glial scar, which blocks axons from growing past the site of injury. Without therapies that aim to restore axonal growth past the injury site, axonal regeneration and cell replacement therapies would not be too successful. Luckily, chondroitinase ABC and intracellular sigma peptide minimize the effects of the glial scar. You'll learn more about them here. Recall the secondary damage following a spinal cord injury and how it triggers the formation of the glial scar. Astrocytes are not the only cells making the glial scar. Molecules called chondroitin sulfate proteoglycans also surround the site of injury and prevent nerves from growing past that point. These proteoglycans contain sugar chains branching from their protein core. After decades of research, scientists discovered an enzyme that's able to free the sugar chains from the protein core. When glial scars are treated with this enzyme, axons are able to grow past the injury site. The enzyme is called chondroitinase ABC, and it's showing a lot of promise. Lab research found treatment with chondroitinase ABC resulted in regeneration of axons and improved motor function. Chondroitinase ABC, used in combination with cells that can regenerate the cord, such as Schwann cell and olfactory and sheathing cell therapies, provides an even greater restoration of function, suggesting that a cure for spinal cord injury will likely be a result of a combination of therapies. Although there's been extensive lab research on chondroitinase ABC, it is yet to reach clinical trials in humans. Difficulties with translating chondroitinase ABC to clinical trials include complications delivering the enzyme in a way that it can reach the spinal cord. Despite these complications, research is being done to translate chondroitinase ABC to clinical trials and to create a virus that will act as a vehicle to deliver a gene into cells, which will then be able to produce chondroitinase ABC. The glial scar is a wall around the site of injury, which traps axons from moving past it. The tips of axons have a receptor that gets caught in the proteoglycan molecules within the glial scar. Picture it as Velcro. The axons get hooked on the proteoglycans and are unable to move. To the axons, they make a synapse-like connection, so they're happy to be there even though they're in the wrong place. A peptide, which is a fragment of a protein called intracellular sigma peptide, blocks this receptor on the axon, allowing it to slide right past the glial scar. This work has been done in a lab. Often though, things seen in the lab are not replicated very well in animals or humans. However, when ISP was injected into mice with a thoracic spinal cord injury, the axon slid right past the glial scar. When this happened, it came with restoration of function, but also with lots of variability. This variability is likely due to sprouting of axons that naturally occurs under the injury site. Sprouting happens from intact axons below the injury site that make new connections. ISP allows axons from above the injury site to slide past the scar and make connections with these newly sprouted axons. Some mice may not have had as much sprouting, so ISP did not help them, while others may have had an extensive amount of sprouting and therefore recovered a significant amount of function. Like chondroitinase ABC, ISP can be given to someone with a chronic spinal cord injury and still likely have an effect. There still needs to be some tweaks with dosage and how to control the sprouting before reaching human trials. However, these early lab studies suggest that ISP gets into the spinal cord and has a positive effect.